Okay, so to kind of summarize the 7 steps for gene regulation, okay, you have the transcription through the DNA packing, the initiation of transcription with the transcription factors. Oops, let me go back. So post-transcription through the mRNA processing, splicing, and the breakdown of the interference RNA. And then you can have translation, so you block the start of it. Post-translational is where you, how the protein is processed, and then also the actual protein breakdown or degradation with ubiquitin. So here's kind of a video to show you how RNA interference kind of interacts with your mRNA during translation. So you can watch that on your own later. So how gene regulation determines cell differentiation and how the organism eventually develops. So this is evolution and conservation in action right here. So there's this dude, Dr. Ed Lewis. He researched fruit flies with legs where antennae should have been. So basically what he realized happened was that one gene didn't produce protein. So there's no antenna and legs developed instead. So the specific gene for antenna didn't produce the protein that was necessary for it to actually be expressed. So the location was acting like another body region. So each gene that controls body parts has about 180 nucleotides conserved in common. Okay, he found this called the homeobox region. Okay, so if you see here, there's a variable region, so the region that can vary, okay, and then you have a homeobox region, which is about 180 base pairs, which equals 60 amino acids. So all genes within that region were called homeotic genes or Hox genes, okay. So Hox genes and cell differentiation, this is so you can kind of see like the placement for like a fruit fly and then for a mouse for your different Hox genes. This is for a human, okay? So this is what it looked like for the fly. Normally, this is where the antenna would be, okay, right here, because this is an eye and this is an eye. And then right here is where the antenna would normally develop, but then here you can see it was actually legs that developed. So then in a dragonfly, you can have the 2 or the 4 wings versus actually having 8 wings. Okay, so that's where we have a mutation that causes more wings than normal. And then here you can see the legs coming out instead of antenna. So here, this is fruit fly, this is the human. Okay, so these are the genes that kind of coincide in both humans and flies. So you can see that the Hox genes are conserved and are variable throughout different species. So Hox gene expression pattern defines the development of all animals, and the pattern of these genes is constant for all phyla. Okay, so this shows evidence for a common ancestor. Okay, so if you look at the Hox gene, if you want to do more research and everything, it's really, really cool and awesome that, like, you know, all these different, Bear, you're fine, that all these different, like, organisms can have, like, very similar genes, and it's all controlled by this area. So here's just some more examples. So you can see, obviously there's different Hox genes. So you have Hox C6 and C8 looked at here between a python and a chicken. So you can kind of see the similarities. So some more examples. This is kind of to show a possible like ancestor between the fruit fly and then mouse. And this is an Amphioxus, so like amphibian. Just more examples. Here's some videos that you can watch to kind of explain if you wanted to look more into it. Like I said, it's pretty cool. So now we're going to go over the control of prokaryotic genes. Okay, so that was all eukaryotic. So now we're going to go over prokaryotic. So obviously prokaryotic cells are way simpler. And most prokaryotic cells are bacteria. So bacteria need to respond quickly to changes in their environment because they are so simple. So if they have enough of a product, they need to stop production because, you know, they don't want to waste energy if it's not needed. But how do they stop production? So they stop production of the enzymes that they're using for synthesis. Now, if they find a new food or energy source, they need to utilize it quickly and figure out how can I use this best for my body. So they need to do this because obviously they need to grow, they need to reproduce, and they'll do this by producing enzymes for digestion. So remember our feedback inhibition? Okay, we went over this when we talked about chemical reactions and enzymes and energy. So if you have a product during this feedback inhibition, it acts as an allosteric inhibitor of the first enzyme in the pathway. Okay, so this enzyme at the end ends up acting as an inhibitor here, so then the rest of the process never occurs. But this is a wasteful production of enzymes for prokaryotes because they don't have as much function needed. So what they do differently is instead of blocking the enzyme function, they block the transcription of the genes for all the enzymes in that pathway. So that saves energy by not wasting it on unnecessary protein synthesis. So instead of producing these proteins as enzymes to go back and eventually stop the original enzyme from being produced, they stop the actual transcription so that way it never even gets produced. So that's the difference between prokaryotic and eukaryotic. Prokaryotics try and do this to conserve as much energy as possible. So cells vary the amount of specific enzymes by regulating this gene transcription. So they choose, turn genes on or turn genes off. So for example, to turn genes off, the bacterium has enough tryptophan, which is a neurotransmitter. Then it doesn't need it's an amino acid, then it doesn't need to make enzymes used to build tryptophan. So it's called a repressible operon. Repressible, that means it's stopping, and then operon, the operator. Okay, think of it like that. Now, to turn genes on, an example is that if a bacteria encounters a new sugar, which is like an energy source, for example, lactose, then it needs to start making enzymes used to actually be able to digest lactose. So that's called an inducible operon. So it induces or causes the operation of it. So operons are actually genes grouped together with specific related functions. So example would be enzymes in a metabolic pathway. Okay, so all the enzymes from that feedback inhibition that was on the previous slide, those would be operons. You can have a promoter, like RNA polymerase binding site, and that single promoter controls the transcription of all genes in the operon. So it's transcribed as one unit and a single mRNA is made. You can have an operator, which is the DNA binding site of the repressor protein, okay, that stops. So repressor protein is a repressible operon. That's what binds the DNA, the operator site, and it blocks RNA polymerase, so that means it blocks transcription. So that's how the genes can be turned off. So, for tryptophan process, okay, which is an amino acid, this is a repressible operon. So the synthesis pathway model is here. So when excess tryptophan is present, it's going to bind to the TRP repressor protein, which triggers the repressor to bind to DNA. So it blocks or represses the rest of the transcription. So then you have a conformational change in the repressor protein. If you change the shape, what do? You should all yell at me, change the function. Oh my God, you're so right. Miss Taylor, I miss you so much. Okay, so tryptophan operon. What happens when the tryptophan is present? You don't need to make the enzymes to build it. Okay, so you can actually conserve energy because you don't need to make the building enzymes because it's already there. So, yay. Okay, inducible operon. So let's look at our lactose example. So we find out that lactose is available. We're a bacteria. Heck yeah, let's get some sugar, right? So the digestive pathway model, what happens is when lactose is present, it binds to the lac repressor protein and triggers the repressor to release DNA. So it basically tells it, hey, let go, so that way then we can go ahead and transcribe. So you have a conformational change, boom, changes the function, boom, we can now transcribe. Here's a video that you can watch about lactose operon. Now, when lactose is present, you need to make the lactose digesting enzymes. So it's kind of like the opposite of tryptophan. When tryptophan is present, you don't have to make those enzymes. But when lactose is present, you have to, because you need to break this down, because this is your energy source. So if you have any questions, because I know these are a lot of different words and stuff that we might not be familiar with, please let me know. There's plenty of video links throughout this to help you kind of explain it in other formats. But if you need help, please, please, please let me know. Your AP test loves to ask about operons. So that's all I have for you. Make sure you do your College Board assignment online. It is due next Monday by 11:45 p.m. I will talk to you guys soon. Bye.